today I will be presenting the work that we recently published in the Journal of Immunotherapy of Cancer, which is on selectively targeting myeloid-derived suppressor cells through trail receptor 2 to enhance the efficacy of CAR T cell therapy for the treatment of breast cancer. So breast cancer is the second most common cause of cancer-related deaths in women, and majority of the deaths are caused due to metastasis. So the use of chimeric antigen receptor T cell therapy for the treatment of solid tumors like breast cancer has been very challenging. And this is due to the presence of the suppressive tumor microenvironment that surrounds the tumor. And this tumor microenvironment consists of suppressive cells like the myeloid-derived suppressor cells, which are the MDSCs. Now, these MDSCs are known to suppress the immune response. So they not only suppress the immune response, but they also directly contribute to the tumor growth by promoting angiogenesis and metastasis. And primarily in breast cancer, increased accumulation of MDSCs has been associated with increased disease burden and poor response to immunotherapies. That is why we wanted to target not just the tumor in itself, but also the surrounding tumor microenvironment by specifically targeting MDSCs. So in order to target the tumor, we chose the MUC1 antigen. Now, on normal cells, it is expressed in the hyperglycosylated form, whereas it is highly expressed in the hypoglycosylated form in about 70 to 90% of breast cancers. And it is associated with poor prognosis and increased risk of metastasis. Now, in order to target the MDSCs, we chose the trail receptor 2. This is a death receptor, and it is highly expressed on MDSCs. And engagement of this death receptor on the MDSCs has shown to induce apoptosis in MDSCs. So this is our proposed strategy. Our aim was to create a CAR T cell therapy that has a CAR MUC1 construct that selectively targets MUC1 positive tumor cells. And then we also synthesized a novel TR241BB co-stimulatory receptor that then targets the TR2 receptor on MDSCs and induces apoptosis of these MDSCs. And at the same time, through its 41BB co-stimulatory domain, it is able to provide additional stimulatory signals, allowing the persistence of T cells at the tumor site, and thereby helps in modulating the tumor microenvironment. So the CAR MUC1 construct that we have used was developed in one of the labs where I work, which is at the Center for Cell and Gene Therapy. So the top panel here shows the expression of MUC1 on breast cancer cell lines like BT20 and MDA-MB231, whereas this is our negative control, and it lacks the expression of MUC1. So here in the lower panel, as you can see, we have performed a cytotoxicity assay. So this assay basically gives a readout of the percentage of tumor cells being lysed, either in the presence of non-transduced T cells, which is our control. So these T cells are not transduced with any CAR construct, or we have our CAR MUC1 transduced T cells. And as you can see, that CAR MUC1 is specifically able to kill MUC1 positive tumor cells, but not the MUC1 negative cells. So further, in order to test now the downstream signaling through our co-stimulatory TR241BB construct, we assess the translocation of NF-kappa B, which is a downstream signaling molecule, into the nucleus. And for this purpose, we seeded non-transduced and TR241BB transduced T cells in the presence of either a recombinant TR2 or anti-CD3, anti-CD28, which served as our positive control. And then for our negative control, we have a few uncoated wells. So we incubated these cells in the presence of either recombinant TR2 or our positive control. And we then harvested these cells at different time points and extracted the nuclear fraction and performed an ELISA for NF-kappa B. And what we observed was that recombinant TR2 was able to trigger signaling through the TR241BB transduced T cells, as you can see here, and induce NF-kappa B translocation, which was very similar to what we observed in our positive control. So we then generated our human MDSCs in vitro and performed a flow cytometry to assess their phenotype using a set of different markers that have been commonly used to define MDSCs. And we observed like a high purity of these MDSCs that were positive for CD11B, CD33. They were CD14 positive, so they were of the monocytic lineage, and they had low levels of HLA-DR. We also assessed the levels of trail receptor 2 on the surface of these MDSCs, since we're targeting trail receptor on them. And we did observe a high expression of trail receptor. So further, in order to test whether these MDSCs are functionally suppressive, we performed a suppression assay where we cultured the MDSCs either in the presence or absence of T cells. And then we observed that T cells that were stimulated showed high proliferation, whereas the presence of MDSCs just inhibited the proliferation of these T cells. And it was very similar to what we saw here in our unstimulated T cell controls. Also, the presence of MDSCs dampened the production of interferon gamma. And so we were able to state that the MDSCs that we generated were definitely suppressive, and we further used them in our downstream in vitro assays. So here we have transduced our T cells with the CAR-MUC1 construct, and we obtained a really high transduction efficiency. And then we further performed an in vitro cytotoxicity assay to determine if the CAR-MUC1 T cells were able to kill the tumor cells. So as you can see, here we observed that either we had our T cells cultured in the presence of tumor cells, either with MDSCs or without MDSCs. And we observed that the presence of MDSCs actually inhibited the cytotoxic potential of our CAR-MUC1 transduced T cells, which is what is expected. So further, we have transduced our T cells with another construct, which is our co-stimulatory construct that we generated, which is TR241BB. And again, we got a good transduction efficiency. And here, we have labeled our MDSCs with the chromium for the cytotoxicity assay instead of the tumor cells, because now we want to assess the percentage of MDSC apoptosis. And what we observe is that TR2 alone, or in the presence of the CAR-MUC1 construct, triggered apoptosis in MDSCs. And this apoptosis was slightly enhanced in the presence of CAR-MUC1, but both the constructs did trigger MDSC apoptosis. So finally, since we saw that individually both our constructs and CAR T cells were basically working, We then transduced our T cells sequentially, first with CAR-MUC1 and then with the TR241BB construct. So the CH2 spacer basically stains the CAR-MUC1 construct. And so you can see that we have got a good transduction efficiency. So these cells are double expressing cells that have both our MUC1 and co-stimulatory receptor. And once we perform the cytotoxicity assay, either here we have our tumor cells that have been labeled, and we observe that addition of the TR241BB construct was now able to rescue the cytotoxic potential of our CAR T cells, even in the presence of MDSCs. So having established the efficacy of our CAR T cell therapy in vitro, we now moved on to an in vivo mouse model. So to assess the anti-tumor potential of our CAR T cell therapy in vivo, we developed a xenograft mouse model where we injected a 5-fly luciferase labeled tumor cells, either in the presence or the absence of MDSCs in immunocompromised NSG mice. And we observed that in the presence of MDSCs, the tumor burden was increased. And also there was a significant increase in the tumor volume in the presence of MDSCs, which also led to an increase in the tumor weight. Now, in order to further investigate this marked increase in tumor volume that we observed in the presence of MDSCs, we performed staining for the tumors in order to quantify vascularization through the expression of CD31. So as you can see that the tumors that were co-injected with the MDSCs exhibit increased angiogenesis, as can be quantified through CD31 positive vessels here. Also, since MDSCs are known to recruit fibroblasts at the site of the tumor, we thought that it's possible that there is enhanced accumulation of fibroblasts, which is leading to this dramatic increase in their tumor volume. So we stained the tumors for alpha smooth muscle actin positive cells, which are fibroblasts, and we observed that co-injection of MDSCs along with the tumor cells did indeed recruit several fibroblasts at the tumor site. And so we had an increased influx of fibroblasts in the presence of MDSCs. So to further track the persistence of these MDSCs at the site of the tumor, we then co-injected immunocompromised NSG mice with tumor cells and MDSCs. And mice were then sacrificed at different time points, and the tumors were harvested, and flow cytometry was performed to assess the presence of MDSCs at the tumor site. And as you can see that by day 21 itself, we see that there are no MDSCs observed at the site of the tumor. And this is the quantification for these results. So this was a very important set of data that we got, because based on this data, we were then able to design the treatment regimen for administration of our CAR T cell therapy. And then finally, to test the efficacy of our CAR T cell therapy in vivo, we used the xenograft mouse model that we had already established. And we then administered the CAR T cells to the mice that had established tumors, either in the presence of MDSCs or absence of MDSCs. And we then monitored the T cell trafficking, proliferation, and persistence at the tumor site. So we observed that CAR-MUC1 TR241BB transduced T cells did indeed show an increased T cell proliferation And they also expanded really well at the site of the tumor compared to all the other groups. And this is the quantification for the same data. As you can see that in the presence of the TR241BB construct, they can persist better at the site of the tumor. We also observed that the CAR-MUC1 transduced T cells were able to reduce the tumor volume as compared to CAR-MUC1 transduced T cells in the presence of MDSCs. And introduction of the TR241BB construct was actually able to significantly reduce the tumor volume and also delay the tumor growth as compared to all the other groups. So to conclude, we observed that our CAR T cells are able to eliminate breast cancer cells and in vitro generated functionally suppressive MDSCs. And in our in vivo xenograft model, we observed that the presence of MDSCs led to increased tumor burden and a significant increase in the tumor volume. And also the presence of these MDSCs enhanced angiogenesis and accumulation of fibroblasts at the site of the tumor. And finally, we observed that our CAR T cell therapy was able to enhance the anti-tumor activity and also overcome MDSC suppression. So with this, I would like to thank firstly my PI and my mentor, Dr. Valentina Hoyos, for giving me the opportunity to work on this project and for her constant guidance, and everyone here in the Hoyos lab for all their help with this project. And I would also like to thank several of the center CAGT lab members. So we have a nice center here, Center for Cell and Gene Therapy. And several of these people helped us with this project. And this project would not have been successful without everyone's inputs and guidance. So I'd like to thank everyone. And thank you for the funding as well. So I would like to thank Secret for the funding. And with this, I will be happy to take any questions.
What needs to happen next for us to implement more effective CAR T-cell therapy for breast cancer patients?
Yeah, that's actually a very good question, and it's really relevant. So as you know that although we did observe better tumor control in mice that received our CAR T-cell therapy, all the tumors eventually returned. So we kept observing the mice till day 42, and we saw that the tumors did return. And it is known that adoptively transferred T cells have been known to benefit from a third class of proliferative signals that are derived from immunostimulatory cytokines, which are actually quite scarce in the solid tumor microenvironment. So an appropriate signal 3 actually increases the CAR T cell activation, proliferation, and also the effector functions of the CAR T cells in vivo. So currently what we are doing is we are working on adding cytokine signal 3 to further increase the persistence of the CAR T cells at the tumor site. And that may help a superior tumor clearance before we can actually bring this therapy to the clinic.
And can you maybe speculate how much more time is needed until patients can benefit?
Our first step would be to optimize our CAR T cell therapy by performing some preclinical in vitro and in vivo experiments to determine whether the incorporation of the cytokine signal 3 actually improves the ability of our CAR T cells to eliminate breast cancer. So, and then based on the results of our experiments, we will then move into a clinical trial. And the Center for Cell and Gene Therapy at the Baylor College of Medicine, where I work, is actually an optimal environment for translating CAR T cell therapies to the clinic, because it houses one of the largest cellular and vector GMP facilities in the United States. So we are looking at around, say, probably in the next year or two, we will be able to open a clinical trial. And so our goal would be to first evaluate the safety of these CAR T cells. And simultaneously, we will be collecting samples from the tumor and the blood of patients that receive this therapy. And we will then perform correlative studies to understand if the T cells that we administer are able to actually survive, infiltrate, and eliminate the tumor cells in MDSC. So based on the results from this trial, the preliminary trial, and the correlative studies, we will know how to move forward. So whether to go back to the bench and address issues if we have any, or then we can move into phase 2 or 3 trials.
Dr. Nalawade, thank you so much for speaking on this, and let me wish you, with this further work, all the very best and a great start to the new year. Thank you.
Thank you very much. Thank you.